So this is not Zoom, right? The platform, but it's similar to Zoom, right? It's Soho. You call Soho?
It's Zoho. It's an Indian version of Zoom.
Wow, it's incredible. We don't have that in Thailand. We always use American things. This is what another thing that I like in India. You have your own technologies. You try to invent or develop something that's useful for Indian people, which different from us. We have a mindset of using other people. We don't have a very uh, the thing that we want to make it ourselves. We all import it and use being like a user. So, but we're trying to change that mindset to be more or less like Indian mindset to build our own technologies. I hope that would be passed to our young generations because my generation is too old to change now. So we have to pass this idea to the new generation. Like maybe we have it today for your class. You also have the young student and also young, career, right? Young people in the career part. I'm trying to find a share. Okay, I see it. Share screen. And I have to choose window, right?
Right. It's same as Zoom, sir.
So, right, and then
Yes, it's getting. Yes, done, sir.
okay. So do you see my full screen now?
Done, sir. Correct. It's visible.
No, but I can't see me now. Wait. I'm sorry, this is my first time using this.
No, it's okay, sir. But you are visible
Okay.
and also your screen is visible.
Okay, you see this?
Yes.
Okay. Thanks once again. This is my honor and pleasure to come here to talk to you. You can interrupt me anytime. If you want to ask any questions, don't just wait for the end and then you just raise your hand and ask me anything that you would like me to respond. And the thing that I'm talking today is something like what we are doing in the lab. Actually, I'm from Faculty of Medicine, not science. So what I'm talking today is going to be like disease related. I'm trying to make it as simple as possible because maybe some of you guys are not really doing a basic science, but we are doing a basic science, not a clinical science yet. But we want to transfer the basic knowledge to be a clinical science and useful in the clinical setting or in the hospital, and at the end, applicable to the mankind all over the world. I'm from Chulalongkorn University, which is one of the oldest university in Bangkok, in Thailand, actually. And yeah, like Sujit mentions, I would love to thanks once again. We become a friend, a good friend now. We first met in March, right, in Golakpao, Uttar Pradesh, and for the conference, and then become, very, very connected. And then, yeah, like he said, I really love the Indian cultures, Indian people, because that would be my first time that I really get into the culture and conference and also scientific communities. And yeah, I would love to have, I have some Indian friends, but it make me more into the Thai Indian cultures and understand what you are thinking, what you are doing, and what future you would like to shape the Indians to the world, thing like that. And it was very fascinated and excited to have the exposed to Indians culture in such a good way. And again, once I would love to thanks Sujit for inviting me again to this class. And I hope what I'm going to talk would be the thing that we can share together and then bring the bright future together. And yeah, I'd like to start with the questions, like, because I'm from Faculty of Medicine, so we work a lot on diseases. So if I ask you what disease that you fear the most, you can write on the chat or anything that, or you can raise your hand to to answer if you want. So I also asked this question to Google's or to DeepSeek. So what do you think the answer is? Yeah, the
So
answer
we are
both of
getting on. Sorry, sir.
yeah,
So we have answers in the chat box where we have answers like cancer is the most favorite answer. Uh,
yeah.
Amina
But
has
I
yes,
can see the
yes,
chat.
yes.
Oh,
Specifically, blood cancer. Again, cancer,
oh.
cancer. Cancer seems to be the favorite.
Wow, okay. So maybe, yeah, it's the real, what is it, perception. Everyone's scared of cancer, right? No one want. So imagine if today you go to the doctors and the doctor tell you that, oh, you got infected, you got cancer. So maybe your world would be collapsed in that day, right? Because we know exactly how cancer works and it's gonna kill. I mean, it kill you someday, not so long, because there's no cure. Of course, if you can diagnose at a very early stage in some cancer, there's a chance to get cured, but it's very rare. Most of cancer is still very dangerous. Yeah, cancer is still a very deadly disease. For example, yeah, it's still the top killer in the world. And the how cancer happens in our body is because cancer actually is it's not an it's a cell inside our body, and it's not the alien cell. It's our own cell that's changed from one what we call normal cell or normal type to be something ugly, something different that's worse to human body. This process we call transformation. It's something like you're watching the movie Transformer, that the robot transform to be a car, something like that. Because it's cancer cell, it's something different, totally different from normal. It's that process called transformation. The normal cell will respond to the feedback. We will be able to control the cell birth and the cell death, but cancer cell is not. We can't control cell proliferation or cell divisions, and they are resistant today. So we can't kill it easily. Yeah, that's two important thing of cancer cell. When our cell turns to be cancer, it's very resistant today, and it's immortal. It's quite difficult to kill, and it's, or you can say it hard, very hard to kill. I showed this movie because it's one of my favorite Hollywood star, Hard to Kill. When I was a kid, yeah, I watched. I really like him, but now he's very old. Yeah, and the thing is, cancer cell is very difficult to kill. If you want to know the reason why it's so difficult, we have a knowledge that in cancer cell, there's different, like I said, it's transformed from normal to be a malignant cell. At least there's 14 characteristics or hormone that's different from normal. And these leading to uh, the cancer cell that be able to resistant to that, and it's very difficult to kill. I will not go into detail of each of characteristic, but maybe too much and out of the scope. But in summary, all of these characteristics of phenotype of cancer cell make the cancer cell is very difficult to kill, and it's very resistant to date, and they're not responding to the normal feedback anymore. They try to escape or try to get away from the immune response in the human body and make their own supply of nutrient, of blood, of everything. That's why they start to grow bigger and bigger to be a mass. So that's why we call mass, because tumor is mean mass, and cancer it actually is mean crab. It's like moving like a crab, because when the cell turn cancer, like they after they getting them like a big mass and then try to disseminate from one side to another, what we call metastasis, or they're moving like a crab. They don't stay in only one mass. So that make the cancer is very progressive and it's difficult to kill. But anyway, we need to kill it, because if we can't kill cancer and cancer kill us and we die. So the only way to win cancer cell is try our best to kill the cancer cell or prevent it not to happen from the normal cells. Yeah, and if I ask you, so if you can design the weapons to kill cancer cells, what kind of weapon that you would like to use to kill cancer cells? Are you going to use like atomic bomb to bomb the cancer cell and then it make it gone immediately, a very powerful bomb? Or are you going to use some kind of sniper to take down one by one, but it's very precisely? It takes some time, but you can be sure that only cancer cell can take down. So which one you want to choose? The thing is, if you want to use the bomb, it's gonna be like chemotherapy that we use it today. We inject the chemotherapy drugs in the bloods, and then the drugs circulate for the whole body, and then it destroy everything. It's not only cancer cell. And this is what we call the side effect of cancer treatment or chemotherapies. And we don't want that happen. So that's why it's most of studies, including us, try to find a way to be something like a sniper, make a weapons or the bullets that can target only the cancer cell, but we leave the normal cell unharmed or other cell still not injured by these weapons. And this field we call targeted cancer therapy. This is the field or the area that we are working on. So our idea is quite simple, but it's not very easy to make. So we want to make something like what we call nano carriers. It's a carrier or the particle is very, very small at the nanoscale that we call a nano carrier. And we need to load the drugs inside the carrier in there, and then make this look like a bullet to kill cancer cell. And how we can direct this bullet to the cancer cell? We want to code it with what we call targeting peptides, tumor targeting peptides, the thing that can tell that can be recognized only tumor cell but not normal cell. And now the targeting ligands, it can be peptides or antibodies. So we work on both. You see this, the green one, this is a peptide, and the blue one, this is the antibody that we coated on the nanocarrier or nanoparticles. And then we think this peptide or antibody will go to bind what we call tumor receptors, because tumor cell will be express some protein that's different from the normal cell. So we have to identify what is the tumor receptor in that cancer cell that express a lot, but not in normal cell. And we use that as a target. And then we decide a peptide or antibody that recognize specifically to the proteins. For example, the green one here, we decide a peptide that bind to the protein called NRP1, which express only on tumor cells. And then after this bindings, and then this tumor, this nanocarrier can be uptaking or endocytosed inside the cancer cell. And once it's inside the cancer cell, the drug inside nanocarrier will be released. And then this drug will attack to the tumor cell according to whatever the mechanism of that would be. For example, what we use in our study, we use DOX, you see here. It's called doxorubicin. It's one of the very powerful drugs. So we put it in the particle and then get into the cell and release inside the cell. And then this drug will go attack to the DNA of the cell and kill cancer cell at the end. The process of killing cancer cell we call apoptosis. So once it's induced apoptosis, and then cancer cell will die. And the next, this is the idea that we would love to help our carrier to attack the cancer cell. And the next question is, what material that we are going to use for making nanocarriers. So because if you want to put something inside the human body, the main concerns or the main issue is about biocompatibilities. You have to be sure that thing that you put inside the body is not toxic or is compatible. This can be degraded, there's no immune rejections, it's not toxic to human bodies. So we try to find what kind of material that's very compatible or very inert or friendly to humans' bodies. And at the end, we come up with the protein because our humans' bodies or the cell built up by protein more than half of the other molecules. There's 4 types of biomolecule. We have protein, carbohydrate, nucleic acid, and lipid. And protein is half of the biomolecules in the cell. So and this protein is very compatible to the cells. So that's why we choose to use protein as a carrier for making our particle. And our hypothesis is, yeah, we want to make the nanoparticle and we use the protein. Now we work on 2 types of protein. The first one, the upper one here, is hemoglobin proteins, and the lower one this is the human serum albumin, this albumin proteins. And then we decide to make the particle and load it with the yellow one here. It's a doxorubicin. We put the drug inside the particle, and then we code it. We conjugate with the antibody or the peptides at the lower part to target the tumor receptor inside the cell. Our hypothesis is when we put this into the cell, because the only tumor cell that's express tumor receptor or antigen. In this case, it's P32, the blue one here, and the green one here is NRP1. This is our target antigen or target receptor on tumor cell, which is there's no express in normal cell or in other cell. So once this particle go into the human body or export to the cell, so that means the only cancer cell can recognize this particle and uptaking it inside the particle. And then it's released the drug inside the cell, and then it activates the cell's apoptosis pathway or cell death. And at the end, it's kill the cell, to kill tumor cell. But on the other side, the non-cancer cell, there's no expression of this protein, so the particle cannot get inside the cell, and then just leave these cells unharmed or non-injuries, no injuries. And why do we pick P32 and then RP1 as a tumor receptor? Because we work very close with the team in Estonia, Professor Tambet, the blue jacket in the middle here. He's very nice and generous. He gives us the chemicals and antibodies and also the peptides, that also the cell line that we could take to initiate this project together and then do it in Thailand in the lab. So that's the reason why, because we have the collaborations and we get tools and antibodies from this lab in Estonia. And what kind of cancer that we are interested in? There's 2 types of cancer that we are interested here. The first one is bladder cancer. It's happened in our urinary bladders here. The main risk factor is still cigarette smoking. You have carcinogen inside the cigarette smoke that initiate the cancer or carcinogenesis. And the other one that's one of the prevalent in Thailand is liver cancer. Yeah. It's associated with the viral infection like hepatitis B and also the cirrhosis or alcoholic consumptions. So these 2 cell types that we are working on now in our lab. And then my student did all the experiments. I didn't do it myself. And then I helped the student to take off the projects. And first step is we have to identify the expression of this tumor receptor, NRP1 and P32. in liver cancer cell and also liver tissue. So we have the liver tissues from the patients because we work in the hospital as well. There are some clinicians and also the professors that can access to clinical sample of patient livers, and then we took that and then we stain it to see the expression of these two proteins in the liver tumor. Sorry. As you can see here, we compare between non-cancer here. Do you see it? Let me show you the pointers. Yeah, this is non-cancer cell. No expression or very low expression. The brown one, the brown colors, is mean there's a positive staining or there's expression of this protein in RP1 and P32. So you see when it turn cancer, you see the staining turn brown. That means there's a lot of expression of these tumor antigen in the tumor cells. Similar to here, this is bladder cancer, and on your right side here is a liver cancer. And A and E, this one is a non-cancer or normal cell. There's low expression of this tumor antigen. So in this data, we summarize that. We can conclude that this NLRP1 and P32 protein is overexpressed or it upregulated in bladder cancers and also in liver cancer or hepatocellular carcinoma. And then, because we want to work in the cell culture model first, in vitro first, before we go to in vivo. So my students have to look into the expression of this protein in all cell lines that we have in the lab. He screened all of them that we have, and then at the end, you see he's classified into, because my student, this boy, he's working on P32. So he's looking for the cell that have a very low expression of P32, which is TTC sub and HK2 cell. And we want to work on the positive cell, which is highest expression of P32. And he get HG2, MU7, and also UMAP3 cells here. As you can see here, the green color is very intense. That means the level of expression of this protein is higher than the light green colors here. And after he know that which cell line that he want to work on, and then he synthesize his particle. He used the hemoglobin from cow. Yeah, he go to the slaughterhouse and then he get the blood and isolate the hemoglobins. And we also collaborate with Charlie Days from Berlin University here, Medical Universität of Medicine in Berlin, Germany. And with Professor Hans here, we learned the technology we call co-precipitation techniques to precipitate the hemoglobins and then make the particle like this. And after we get the particle, we load this doxorubicins, and also we coat it with the blue antibodies here. The antibodies with recognized P32 antigens on the tumor receptor. This is the size and the shot of particle. I won't go to the detail of this. Maybe it's too much physics or chemistry here. But anyway, we got the particle that we designed for. Yeah. We have hemoglobin particle, we have the drug inside, and when we coated it with antibody, recognize the P32s. And then what he do is the experiment we call uptaking. So he wanted to know that whether the particle can be uptaken by the cancer cell or not. Like, for example, he monitor for, this is the first columns here, this is 0 hour or at the beginning. At the beginning, after that is 1 hour and 2 hours. You see the intense color of red color here. That means the particle getting taken inside the cell. So we prove that the particle can be uptaken by tumor cells. And another experiment that he make it is quite convincing to me. Yeah, what he did is he co-cultures two cells together. One cell has very high expression of p32, and the other cell is very low expression of p32 here. This is HepG2, and this is TCC subcell. And after he co-cultured this two cell type together, and then he put the particle that he designed and propagated it inside the cell. And as a result, you see here, the HepG2 here, which is higher expression of p32, uptakes a lot of particle. Yeah. You see the green particle here? This is the particle that he put in the cell. But TCC subcell, which is negative for p32, that's very low uptake or endocytose of particle inside the cell. So this data show that our particle is very selectively or very targeting to the tumor cell that express p32 or target receptor, but not the other cell that have low expression of target. And then you want to see whether after the cell eating or getting that particle inside and whether it's induced cell death or apoptosis inside the cell. The experiment is quite complicated, but you just see here the red color. If you see the red color, this means the cell is dying or it's getting apoptosis or the particle still killing the cell. Like you see here, after we put the particle and then the cell getting or eating the particle inside and then the drug release, after that the cell die here. Also here on your left hand or right hand side, he cultured like the tumor mass, tumor spheroid inside the dish. And then as you can see here, the green color, which is indicate the apoptosis cell or dying cell. So after we put the particle inside the cell, in 2 or 4 hours, the cells start to die. Like you see the green fluorescence in the pictures. And that models for liver cell, and then we move to the bladder cancer cell. Again, my student tried to see the expression of this NRP1 and P32 in the tumor cell, in the human tumor tissues. And of course, and then again, we see the overexpression of both antigens in tumor cell of bladder tumor cell tissues. And what she did is she used albumin and to make the particle like this. And again, we do the same idea. We loaded the doxorubicin here inside, and then, but this time we don't use antibody as a targeting ligand. We use the peptide here, the star as you see here, she coating with the peptide, which is smaller. And the idea would be the same. This particle will go and bind to the receptor on the cell, and then get uptake inside the cell and release of the drug, and then kill the cell at the end. And this is the process that she make the particle. She used a technique called dissolution techniques, and she get the particle like this, and then we check whether we have the drug inside the particle. Yes, we get everything that we designed for in the particles. And again, that she worked to test the efficacy of this particle in the cell. She again checked the expression of NRP1, which is the red color here. This is the positive cell. You see the red color on the cell. And the lower part or the lower panel is the cell which negatively expressed, or there's very low expression of NRP1, or target receptor on the cell. And she worked on this both cell. And then first, she see the uptake and the peptide that she coded on the particle called IRGD, which is recognized also in NRP1. And what you see here, compared to the cancer cell, UMAC3, and the normal cell HK2 here, you see the particle get uptaking into the cancer cell much more than normal cells. That we can see the green colors here. And then she did this co-culture techniques to co-culture two cell types together in the same plate. You see here, and like you see here, this is UMAC3, this is the cancer, and M21 here, this is the negative control cell. So when she put the particle inside, you see the particle is green here. So the nanoparticles go only into the positive cell, UMAC3 cell, but not negative cell or control cell, M21 at all. So this data again confirm that our nano carrier can be very precisely target the cancer, but not normal cells. And she did a series of experiments to co-culture between two cell types. And this is UMAC3, which is high expression of target 2 antigens. And this is M21 here. You see the particle, which is green, is go only into the cancer cell, but not the normal cell. And the other side, she also cultured M21 with another cell type of bladder cancer called VM-CUP1. You see the particle likely to go to VM-CUP1, but not M21. So in this data, we can conclude that our particles that very specifically to tumor cell that express the target antigen that we are designed to target to. And again, she's checking for apoptosis or the cell death, activation of the cell death or killing the cell, right? After she put the particle in, again, like you see the red colors here, that means the cell is dying, getting the dead cell pathway getting activated, which means our particle can kill the cell effectively after it get uptaken to the cell. And the other experiment that we do it in vitro is one of my technicians also designed to do what we call colony formation assay. We culture the cell to make the colony. M21 is negative cell, VM-CUP1 positive cell. Okay? So if we use only the doxorubicin like this, it's gonna be like chemotherapy. It's because doxorubicin is very powerful drug. It can kill everything that's get contact with. You see here in the middle columns here. So doxorubicin can kill both M221 and VM-CUP1 completely because this drug is very powerful. It's like a killing agent. So it died both. And when we use our nano carrier or nanoparticles, you see here, it kills only the tumor cell that have expressing the tumor receptor, but they spare the negative cell or the normal cell at the upper panels here. So in this data, we think our particle, it works like a sniper. We can choose the cell or the tumor cell that we want to kill, and we can leave unharmed the normal cell or the cell that we don't want to target or that we don't want to kill. So in this way, we can minimize or reduce the side effect of the drug, and we can be sure that we specifically or selectively target only the tumor cell. Yeah, that thing that I would like to share to you today. Yes, it's still the in vitro study, and then we're gonna move to the in vivo in the animal after this. So I show you that we can successfully synthesize the protein particle from hemoglobin and also albumin. And we can code it with the targeting ligands. In our case, it's antibodies and peptides. And then I show you that we work on the cell culture models that we can convince that our particle can selectively uptake by the tumor cell, but not normal cell. And then it's also activate the cell death pathway or apoptosis pathway in tumor cell that uptaking the nanoparticle. So in this way, we show that our nanoparticles effectively and selectively kill only cancer cells, leave the normal cell safe or unharmed. Yeah, and that the thing that's for academic or research thing that I would love to share. And that's something that I like to share before I'm ending my talk. So, because I know you are still the young people, the young generation, or you you're starting to start a new career because you are early professions people to start your job or your career part. And the thing is, because in the future you don't know what's gonna happen, but I can tell you as I have been through in that phase already. So because now you're selling in the oceans, you have to be independent. You have to learn new things, a lot of things that's come to you. And you can't choose what you have to learn. You can't choose what is going to come to you. It's like you are sailing in the sea, in the big oceans now. But you have to keep in the positive way. You have to have a very positive mindset to go through that, your career part, your life, the thing. And what I'm saying is you can't stop the wave or the problems or the obstacle or whatever that's come to you right, or come in between your life. What you can do is you can learn from it. You can learn how to serve and learn how to survive, how you learn how to get through that. And then every time you learn and you get more skill and skill, and then you get stronger and stronger. That's how life works, how things gonna be. So don't get discouraged or disappointed by things that destroy you. Don't let things that come to you to kill you. Just learn from them. Let them to teach you to be a better person, to be a better, stronger one, and then you will get what you want at the end. Get through that. And remember, a smooth sea never made a skill seller. If you want to have a skill, you do need to confront it and sell your life through the big oceans of yours. Never, yeah, if you want to get the skill, you're gonna get a lot of scar. That's true. But you're gonna be a very talented and a very skillful person after that. Yes, and also remember that you have to keep learning and don't give up easily. This is the key thing that you need to have if you want to be a success in some way, in what way, in any way that you would love to have. It depends on, I don't know what goal of you each person. Everyone have their ambitions, everyone have goals. But what I'm saying is, I'm sure that you will get disappointed, you will get failure, you will get rejection many, many times. But the key is to keep learning from that. Don't regret too much on or resilience, very important thing. Don't give up. And every time like this, I really like this word again, right? AGAIN, again. If you fail this time, you get rejected this time, you have to try it again, right? Don't give up yet. You have to try it first. And before you're trying, you have to learn from the failure of the first failure. Try again, and then you get learning more and more. I cannot tell you how many times you have to do it again. It depends on how much you learn from that failures. And again, until finally you can knock the A out. Yeah. And at the end, you will get GAIN. You will gain what you want. You see, the key thing is you learn from it. Learn from the failure, learn from the rejection. Don't get up easily. Yeah. I mean, after you fail, you have to tell yourself to get up and walk forward, do things forward, learn from it. And at the end, you will gain what you want. Yeah, and thank you very much for staying with us, and thanks Sujit again for having me today. And I also would like to thank my university, my organization, and the grant that give some money to support our research work, and all my friends from Europe, from America, and also from everywhere around the world. Most importantly, I would love to thank my students, because without them, I don't have any data to show you today. So because I cannot do the lab myself anymore. Yes, I have to thank all my graduate students, master degree, PhD students, and also my colleagues and friends. Also to you, Sujit, thank you very much. I
Yes.
would love to have
One question is there. We'll first
question.
take a question. Rajan has written, what type of techniques did you employ in your studies?
Which techniques? I mean,
Any specific technique that you used in your research activity?
oh, we use a lot of techniques. Most of them are molecular biology techniques like qPCR, like to see the cell cultures, to identify the protein expression like immunohistains, and also the Western blottings. But to summarize, it's all about biochemistry techniques and also molecular biology techniques.
Rajan, you want to ask for any specific technique? You want to hear from him?
It's fine. Yeah, it was a general question. I was looking at the pictures. I was really amazed with your
Oh, thank
photographs
you.
and their clarity. Yeah, I have been in the lab. I can understand how difficult it is to capture such clear images. So really, I
Right.
congratulate your amazing work. Yeah.
In that image, we use confocal imaging. Yeah, if you want to have a very good image, clear and contrast, you need a very high resolution microscope. The highest one now is confocal microscope. Yeah, we use confocal fluorescent
Exactly,
microscope.
because your pictures were so clear, and that's how I was looking at the
Yeah,
techniques. Thank you very much.
Yeah, totally. Thank for appreciating our pictures.
Any more questions? Okay, we'll wind up, Dr. Chanchai. I'm really thankful to you for accepting my invitation and for making it such a interesting topic. We come up with these topics every Saturday, and we have a community where we have almost 1,000 people in there. And depending on their topic or their interest, they join these sessions. We try to bring different topics from a broader pharmaceutical perspective. And as you rightly captured in that last slide, with respect to, of course, everything with respect to your research is amazing. I have had the honor of hearing you 2 to 3 times now, but with respect to, I will be speaking only with respect to the last slide. The last slide which you showed.
The again slide, right?
Yes, that slide with respect to the gain means again and again, as you slightly said, because I feel that we all know, as industry person or as researchers, know that these are times of uncertainties, with especially with new technologies coming up like AI and all coming as a what you call as a breakthrough, but as they are creating uncertainties in the job market also. With respect to research also, many things are changing with respect to the way you approach basic things like writing a research paper itself, or the way you are conducting research. Everything is changing. So this, I can say that this is uncertainty with respect to even your fundings also. There is no clarity on certain aspects. So just because one aspect didn't work out, you cannot stop there. If we are stopping there, then there is no research. You have to search again. That is research. So you have to keep working there, and that is the main aim with which we are connected. That we are trying to bring experts who have already gone through the pain. the gain is there, that pain part also is there. So that they have gone through that pain, and you are seeing them as experts because they have suffered that. And by hearing their efforts, more than their research, it is their endurance, what you call as their perseverance, the motivation to come to the lab every day in spite of the failures. That's what they have made them the experts. That is the one lesson that each one of us has to take away from, I guess, from Dr. Chanchai also. I know he would definitely have his own story of personal struggles. We are seeing a well accomplished or a person who has 4 to 5 patents and 60, 70 research writings or on in top class journals. But to achieve that, I guess, I know he would have his own stories with respect to the whatever struggle he had with respect to being a researcher. So that's something we have to learn from him. And I am especially thankful to each one of you, Dr. Chanchai. Of course, that's the first thank you. The others also for joining in, for putting up their questions, taking out time. As I always say, more than any money or anything, the time factor. You are giving that 1 hour to me, or that trust you had in the session. Hope we have been able to justify it. And we will definitely come up with more interesting topics like this, and we will try to have as many as speakers. We will again get back Dr. Chanchai soon, and I know he will be graceful, means he will definitely accept it. He's a good professor with respect to teaching also. He has this good knack of teaching harder topics in a very simple manner. So thank you so much.
Thank you
for connecting. I'll always also be uploading this on YouTube, so you can see that video there also, in case you want to have any references or with respect to, of course, if you have any specific questions for Dr. Chanchai from your research field also, you can connect with me. I will just pass on the questions to him if you are not asked this. Okay, thank you so much, each one of you. Have a great weekend, and let's hope for a great vacation season is coming up. So we will see each one of you after the vacations. Now we'll break because everyone will be busy with their own of course Christmas is there, then New Year celebrations. So we'll take a break with respect to the learning part. Just energize yourself and start afresh in the new year 2026. Dr. Chanchai, wishing you a very happy new year ahead, and may you bring more and more great researchers into this field, and hope you have a great year again. Okay. See you soon, guys, all of you.
See you.
Bye bye, everyone.
Merry Christmas and Happy New Year to everyone. Thank you.
Bye-bye. Thank you so much.